The next one, third one is the in situ methods. What is meant by in situ? In situ is nothing but the particular location or inside the location. Here in situ means the tissue level study. Tissue level study is nothing but as it is in their physiological conditions we are keeping, in that only we are giving our trial drug. This is called as a in situ methods. Here in situ method is conducted on organs or tissue in their natural position to maintain the physiological structures. That is as it is, how it is the naturally present in the human beings. Similarly, they are separating and they are keeping in their natural the positions and maintaining the physiological structure. After that only we are doing the research. This is the in situ methods. In situ is nothing but their location, inside the location, as it is the locations we are doing experiment here. The example is isolated rat intestine used to study the effect of the eagle marmulose extract on the intestinal motility. That is bilwa, that is eagle marmulose. Bale patra is the common drug we are using for the loose motion or diarrhea, dysentery. In the Ayurvedic, commonly they are using Ayurvedic physicians in the loose motions to show their property, whether the drug is acting or not. That if you want to prove, you can use this in situ methods. What here the experiment is that isolated rat intestine. First of all, that rat intestine we have to separate. That intestines as it is to physiological structure, the natural position used study. In that intestine we have to add the bilwa, eagle marmulus extract on intestine, whether its intestinal motility is there any changes or not, that we are analyzing. This is the one of the very good example of this in situ method here in the preclinical methods. Next is the application where this in situ method is we are using. The first is in situ hybridization. Here in situ hybridization, here in this the gene analysis, where the gene is located in their natural position, that also we can analyze in this method. So next is the immunohistochemistry. In this the proteins locations we are capturing by these methods. Next, third is isolated organ perforations. It means that the separate isolated organs we are separating and doing experiment or doing research. This is one of the advantages. These are the advantages or applications of this in situ methods. Next one is the benefits or advantages. Benefit is more physiological relevant than the in vitro, because as it is the physiological conditions, in that only we are doing the experiments. That's why it is one of the very good advantage, more physiological relevant than the in vitro. And limitation, what are the limitations of this in situ method here? Technically demanding. That is, the technical demanding is nothing but to do this technique in situ, the expert technician requires. All doctors or researchers can't do this experiment. The special technician requires. This is one of the limitation. Next one is the partial system views. Only the partial system view, because the only one time, this is the example rat, in that only we are separating the intestine to check whether in the antidiarrheal property or it is there or not. That only in the specific organ only we are checking. This is the partial system view only we can check here. Okay, that's why these are the limitations are there about this in situ method.